Hello everyone, my name is Raissa Krupek. I'm a member of the GVENCK group and we are here today at the 9th International Meeting on Plant Breeding with Professor Zhao-Bang Zeng. Hello Professor Zeng.
Hello Raissa.
Dr. Zeng is a professor at the North Carolina State University and is a leading expertise on theoretical and statistical quantitative genetics. So Professor Zeng, tell us a little bit about your career path.
That's a very interesting story that I went to Edinburgh 1983, Department of Genetics to study quantitative genetics. My advisor is Bill Hill. Yeah, he's a very well-known figure in quantum genetics. I stayed there for 3 years, finished PhD. That was actually life-changing experience, really, and I learned quantum genetics, really actually laid good foundations. 86, I came to North Carolina State University, started to do a postdoc with Professor Clark Coker. He's another actually major figure in quantum genetics. So I was so fortunate to really learn from two masters in quantum genetics. That's really a great experience. So I stay 4 years as postdoc, and then I stay on the faculties and stay there ever since. About 39 years in North Carolina State Universities. A long time.
Yeah, it is a long time.
Yeah, that's right. Actually, my training was quantum genetics. I did the theoretical, statistical side of the things. I didn't do experiment. That in the 90s, when the genomics came about, so we tried to learn genomic data and try to use that one to map quantum trend loci. So that was my main research interest during the time in the 90s. That is when I made quite a bit of influential contributions on the statistical method to map QTLs and then to go from there.
Wow, that's interesting. Thank you so much for sharing it
Yes.
with us. Okay, so can we go to the next question?
Sure.
Okay.
Go ahead.
So much of your work involves QTL mapping in complex and polyploid populations. Could you please explain in simple terms how this mapping works and why polyploid populations present an additional challenge?
That's a very interesting question. About 10 years ago, the Gates Foundation, they were funding a big project on sweet potatoes, primarily supporting the Africa's agriculture. Then they feel the need to add a component on genomics. So I was asked to join the project. Before that one, actually, most of my research on diploid, a human and most of animal plants were diploid. There are two copies, one from mom, one from dad. But sweet potato is called a hexaploid. There are 6 copies. So that individual inherited 3 from mom and 3 from dad, so to speak. It was very difficult. The difficulty is purely because of the combinatorial sense. So if you inherit 1 from mom, 1 from dad, you got 2 possibilities because mom and dad each got 2 copies. You got to get 1 of it. So at each so-called locus, 2 by 2, there are 4 combinations. Whereas hexaploid, there are 6 copies of each parent. They're going to inherit of any 3 to the next individuals. So that you have draw 3 out of 6. There are 20 possibilities from mom and 20 possibilities from dad. 20 by 20, 400 possibilities. So at each local, you got 400 possibilities, but we got genome. How many base pairs? Human, we got 3 billion nucleotide base pairs. So that to figure out the transmission from parents to offspring, because eventually we want to figure out for each offspring at each locus where it came from each parent. That's our job. It's very, very hard. The data we're using is called SMP, single nucleotide polymorphism, that is bioanalytic. There are two forms. Fortunately, we have technology can get millions of this locals segregated binary. But our job is locate of individual whole genome and the whole populations and to figure out at each pieces where it came from. So statistical challenging. Actually, it's very well known that this problem is one of the most difficult problems in statistical genetics. That's what we did. Fortunately, I had two very talented students from these universities, Masala Molinari and Guilherme Pereira. They are the one, actually, they work on a project for quite some time. They solve the problem. They solve the problem not only actually the problem is from the millions of pieces of a number of individuals and their parents, and they figured out that we want to construct a so-called complete linkage map. Linkage map is a string of those pieces. They inherit together. And then build a map, and also to infer the haplotype. Haplotype that at each chromosomes, there are 6 pieces. Which segment come from which parent, which segment. That's what the haplotype construction. So we develop the software tools called map poly. We had version 1, now we had version 2. Really can reconstruct the complete linkage map with haplotype inferences. And we also have another tools called QTL poly to map QTL, quantitative trait loci, that how it's segregated from population, how they're responsible for the disease resistance, for the yield, because we want to use that one for the crop improvement. I'm a statistician, I'm a geneticist, I'm a statistical geneticist. So that's what we do. We develop tools, help breeders to solve problems in breeding.
Awesome, Professor Zeng. Thank you so much. So the next question that we all want to know, looking ahead, which areas do you believe that will be the most promising or deserve more investment in the next 5 or 10 years in quantitative genetics and biostatistics applied to plant breeding?
That's actually a very interesting question and also very challenging question. I can tell you why. From my point of view, where last 30 years, we have been going through a so-called genomic revolution. Genomic technology brings so much information to us. We have been cracking the information, really actually turning into the usage for plant breeding, for many other uses. There are still a lot of challenges from that one. One of the challenges is linked genomics with, in the conference, other omics data that not only try to link the genes or contribution loci to a phenotype, but also try to understand the connection with other data, that RNA-6 data and proteomic data, metabolite data, really try to understand the so-called functional pathway. That's one thing that's called system genetics. Another thing is the high-through phenotyping tools, the drones data. We will try to make the automations. So that's actually another major topics in the conference people are talking about. But the major impact change coming is AI. No doubt. AI, it has already actually made dramatic changes, that particularly the AlphaFold, the alpha genomics. Those really bring very rich information. It very soon they try to integrate essentially the most of all of information is already in the databases and then synthesize it in the way people can use that one. Definitely AI will bring us a lot of information, and for the new student and for the newcomers, they absolutely need to understand it, use it. That the challenge is that AI will be a wonderful tool, but it also going to change the way we're doing the research. How it's going to change is still remain to be seen. So that's obviously, I'm sure most people feel a little concerned about it and excited about it as well. And yeah, I will actually encourage the students really dive into those AI tools, so essentially to find out what you can find out there and to make adjustment on your research.
Certainly, it's very important.
Absolutely.
And what were your initial motivations for choosing statistical genetics as your field of expertise?
It was, to me, it was actually a very interesting story. I like mathematics, even in high schools. But after graduate high school, it was during a cultural revolution in China in the 70s. University was closed. So we didn't have a university to go into that. So we were sent to countryside to, that's since I was city boy, but sent to spend 2.5 years in the countryside. And then university reopened in 1977. So we were the first class to admitted to university after 10 years gap. When we got to university, actually, to be honest, it was a chaotic experience that I wanted to go to the university to study mathematics. But for some reason, I was assigned to agriculture university to study animal science. So yeah, there was kind of this so-called randomness going on that way. So I went to the Huazhong Agriculture University to study animal science. And when I actually come to a course called genetics, I was interested in it. Why? Because it has mathematics. Because I like mathematics. So that's why that I say, okay, I'm going to do genetics to employ my mathematical skill. So when I had a chance to go to Edinburgh to study, 1983, so I'm going to choose to quantum genetics. And it was, for me, mostly due to my interest in mathematics and then get into the genetics and later on genomics. I really love it. So I've been over 40-some years, and I made my contribution, and I think I have already lived a wonderful life. So I'm still continuing to do it. And partly it was an accident, partly it was driven by your own interest, and choose the right path along the way. And I feel I'm fortunate. The fortunate in sense I had a profession, which I love it. That's the best way. Combine the interest, the hobby with your profession. What a life you can ask.
Certainly.
Yeah.
Awesome, Professor Zeng. And as a professor, how do you see the challenge of training the next generation of researchers in these areas, and which skills in statistical, computational, and biological should be prioritized in the graduation school?
Our field is called interdisciplinary. It's interdisciplinary because our research subject is biology, genetics, genomics, and behind that one is evolutions, history of the population. So students definitely need to understand that appreciation of our research subject, the question. So that's the first thing I would like a student to understand your subject. You really, and of course, when you start that one, we are the other side is mathematics, statistics, and computations. You need to have a very strong skill. You need to have strong skills to understand data, because we are pretty much is the data science, it's the genetics, dealing with genetic data. You must have understanding the logics behind the data. Statistics is the data of science. Data come with error. So you need to understand where the error come from. Behind that is probabilities. That's a branch of mathematics. So you should have very solid foundation of probabilities, statistics, the framework to deal with data. That's actually very important part. The other part, you should have a strong computational skills to manage data, to analyze data. And there's so much computation tools developed. You should really be able to manage it. Where in the so-called big data era, and genomics come with enormous amount of data, you must have ability to really do large data analysis. So those are the three components for us to train students. That, but always keep in mind that one. The statistics, computations serve to answer biological questions, so that your impact of research will inevitably determined by whether you answer the question in the right way or in the wrong way sometimes. So that to have actually very ground understanding biologies, very strong understanding of data structures, and then the theory behind those things, and abilities to manage analyze data. Those are three components. It is very challenging for any student.
It is.
Yeah, a student sometimes are strong in some side, either in computational side, quantitative side, or sometimes in biological side. But you need to try to make up other part as well.
Okay. So, as your last question, what legacy do you hope to leave, both as researcher and as a mentor within quantitative genetics?
That's a very interesting question. Actually, as quantitative genetics, I'm actually trained as a quantitative genetics from beginning until the end. So that over the years, particularly in the recent years, with accumulated research experience and thinking through my field, quantitative genetics, I now have a kind of understanding that one is quantitative genetics deal with the genetics of quantitative traits. Quantitative trait essentially is any biological features we're interested. We're interested because of, for human, is diseases. That's a quantitative trait. For agriculture, is yield. We want to improve it. Or disease resistant, we want to make a change of it. If we study evolutions, the phenotype, the fitness. Because natural selection operates on the phenotype. So that's sense. So we want to understand the genetic. What is genetic? Multiple genes. We want to understand how the multiple genes working together impact a trait in the population. That's the core of our subject. In the past, we don't have the means to deal with it. We don't have genome informations. But in the last 30 years, we started accumulating genome information, so now we have the means. In the 90s or 2000, population data were limited. So our goal of analysis is somewhat limited, which is to find the QTL one by one. But now, in the last 10 or 15 some years, we have very large data. We have very large complex population. In my opinion, we now have the means. We now have the information in the data. We also have the means to study it. We should get our focus to study the whole totalities of the relationship, multiple gene to a trait. So we're in the golden period. That, to be frank, that I feel currently our discipline has been gone a little astray. They are using the marker not in the informative, but they use marker for predictions, for finding QTL one by one, or GROD, GBLOB. That can achieve a certain goal, but we can do more. The more is about we can use this data to infer a cohesive genetic model, explaining the function connecting the genome to the phenotype or phenome. Use that one to ask the basic biological questions. That is what I've been doing in the last few years. That is actually my yesterday's talk. I want to send the community a messages. Now we have the information in the data now. We just need to get our focus, get our mind into that basic question. I think finally we now at the gate answered basic biological questions after almost 2 centuries now. The biology, we try to understand, observe natures, we observe what happened, we design experiment. We collected now genomic data. We are at the time can put it all together. That's basically what I'm trying to persuade the community, concentrate on this problem.
Perfect. Well, unfortunately, Professor Zeng, we are already in the end of our interview, but I would like to thank you very much, not just for attending this meeting with us, but also for sharing your words with us. So thank you very much.
Thank you. Thank you for you giving me time to have my opinions expressed.
We hope to see you again in future editions.
Thank you.